Good morning everyone. Audio video clear? How are you students? I hope all of you are doing well and you are enjoying your holidays. Good morning. Good morning. Okay. Audio video clear? Okay students. So in last class we discussed about work done. How to find out work done, that we discussed. So we discussed that we can calculate work done by two methods. One is area of the pressure volume graph, when to use this concept if graph is given, and another is integration method. You have to put the pressure value, then you have to integrate, then you will get answer. Okay, so here we solve some questions which are based on this work done. We solved these questions. Okay, so some concept that you have to remember. Here also we solved this question. This question is, here you can see this complete process is cyclic, right? But here in this question this process is not cyclic, है ना? It started from A and at B. It is not cyclic, right? It is linear. Okay, so for both process I explained you how to find out work done. I explained you, if you remember, again we can write, if closed process is not given, if closed loop is not given, suppose given graph is like this is pressure and this is volume, graph is like this. Okay, so what to do? So I explained you have to join the starting point and end point with the volume axis. Okay, joined. Then calculate the complete enclosed area. Calculate the complete enclosed area. This area you have to find out. This area. This area. Is it clear? Understood? So this complete loop area we have to calculate. If process is given from A to B, then what you have to do, join the starting point with volume axis, final point with volume axis, and find the complete area of this closed loop. In the question it was not closed loop, it is AB straight line. We make it closed loop by joining the starting and final point with volume axis. Okay, now next type of question, if closed loop is given, then what you have to do, closed loop can be understand this particular concept. Suppose it start from A, okay, then B. Process is like this, A to B, means arrow sign is given, we are moving from A to B, then B to C, okay, then C to A. Agree or not? So we are moving A to B, then B to C, then C to A, right? This is a loop. Now no need to join starting point and final point with the volume axis. If loop is given, if closed loop is given, then only we have to find out area of this closed loop. Here it was not closed loop. If closed loop is not given, process is only A to B, then what you have to do? Then join initial and final point with volume axis. Join initial and final point with volume axis. and calculate area. Here complete loop is given. Complete loop is given, then its area is equal to work done. Is it clear? So here directly we have to calculate its area. So this area we have to find out. This area we have to find out. Right? Now, here understand two type of question. This is cyclic process. Cyclic process means complete loop is there. Cycle is there. Okay. We can say that this is what cyclic process. Now, this cyclic process can be given in different way also. Understand? This cyclic process can be given like this also. Pressure, volume, same triangle same triangle closed loop I am forming. You can see here, but it is like this: A to B, this is A, this is B, then B to C, this is C, to B to C, then C to A. Can you say what is the difference between this diagram, this cyclic process and this cyclic process? What is the difference? Send your response, I am waiting. Send your response. What is the difference between this cyclic process and this cyclic process? Here you can see this is triangle. This is also triangle. But what is the difference between them? Can you observe here difference between both of the cyclic process? Yes, very good. Jahanvi, this girl is intelligent. You know, this is online class, right? And in online class also, she compelled to identify herself, right? Because most of the time this girl is sending correct answer and fastly she sent. Very good. Other are also correct. You are saying correct that this is anti-clockwise, right? And this is what? This is clockwise process, right or not? So we understood that work done is equal to area of the loop. Here also work done is equal to area of the loop, but process are different. Here it is anti-clockwise and here it is clockwise. Agree or not? Here we can see that this volume increases, then volume decreases. Here we can see first volume remain constant, then volume increases and then volume decreases. Okay. So both are different process, because clockwise, anti-clockwise also tell us about that how process is proceeding. Okay, so what you have to remember here, very simple point, in all cyclic process, work done is equal to area of the loop, whether it is clockwise or anti-clockwise. Now, what clockwise and anti-clockwise, where they matter? So, in clockwise process, work done is positive. Here this is important point. In clockwise process, work done is positive. Why it is positive? Why it is positive? Because here volume increases. Ultimately in the process here volume increases. Okay. Volume increases, then work done is more, positive. Positive value is more. Volume decreases, then negative work done is there. You know, work done formula is PdV, right? dV is change in volume. If volume decreases, then work done negative. If volume increases, then dV means change. Change means final minus initial. If volume increases, the final is more than initial. So difference final minus initial will be positive if volume increases. So if volume increases, then work done is positive. So here volume increases. So in BC path work done is more. C to A volume decreases. But here work done is less. It is negative but less negative. Here it is positive but more positive. So if we add them, so ultimately we will get positive. Is it clear? Here understand. Here A to B volume increases. Okay. Here volume. Understand this point once again. A to B volume increases. Volume increases, then work done positive. Okay. So here work done positive. Then B to C volume decreases. If we are moving in this side, to volume decrease. B to C, to here volume less, here volume more. Okay. So B to C volume decreases. Okay. So here work done negative, but this negative work done is more. This negative work done is more because for this part work done is more. Why? Because pressure is more. Right or not? B to C, in B to C process work done is more, but it is negative. A to B process work done is less, but it is positive. So positive and negative, if we add them, so finally what we will get? Negative. Why? Because negative value is more. C to A, no change in volume. If no change in volume, then work done 0. Why? Because work done formula PdV integration. dV change in volume. No change in volume here because complete volume is constant. So here work done 0. Here work done positive. Here work done negative. You can write this point also. W A to B work done positive. Then W B to C negative. Then W C to A 0. But this negative is more. Why? Because for this part pressure is more and work done is multiply of pressure with change in volume. You can see this is above. Above means pressure value is more. This axis will give pressure. Pressure value is more. So pressure multiplied by change in volume. So it will give more value, but it is negative. So if we add them, so finally we will get negative. So for anti-clockwise process work done is negative. So very important, in anti-clockwise process work done is negative. Clear? In anti-clockwise process work done is negative. Understood? So here work done negative, here work done positive. Again understand here how we can say positive. Can you tell how much work done for A to B process? Send your response. How much work done for A to B process? How much work done for A to B process? Send your response. Yes, very good. Ratna 0. Very good. Then Lekhya 0. Very good. Very good. Very good. Most of them are saying 0. Yes, correct. A to B volume is constant. Here we can say that A to B for this part volume is not changing. If volume is not changing, then work done is 0. Why? Because work done is equal to integration of P dV. dV is change in volume. Okay, very good. All are sending correct. Now B to C, can you tell B to C work done positive or negative? Send your response. B to C work done positive or negative? B to C work done positive or negative? Very good. Again, negative, negative. Yes, some are saying positive. Okay, here you done mistake. Some are saying positive and some are saying negative. Those who are saying negative, their answer is wrong, and those who are saying positive, their answer is correct. Understand? B to C volume increases or decreases? So B to C, this is B point and this is C point. So initial volume, this is initial volume, right? Final point C to final volume, this is final volume. To final volume, initial volume, which is more? Final is more. Means volume increases. If volume increases, then work done positive. So here it is positive. Now C to A, you can say all. You all can say that it is direct. Here work done is negative. So W work done C to A is negative. Why it is negative? Because C to A, final is A, initial is C, final volume is less, initial volume is more, means volume decrease. If volume decrease, then work done negative. Okay, is it clear? This point you all have to remember, that if volume increases, then work done positive, volume decreases, then work done negative. So in all cyclic closed process, work done is equal to area of this loop. Here also work done equal to area of this loop. Okay, so in all cyclic process, work done equal to area of the loop. Okay, and in clockwise positive sign and in anti-clockwise negative sign you have to take. Is it clear? Understood? Now I am giving you a question, you have to tell the answer. Okay, and yesterday we discussed about this work done, everything we discuss here. So here you can write this particular point. What? If volume increases, then work done increases, then work done positive. Work done positive. And if volume decreases, if volume decreases, then work done negative. Volume constant, then work is 0. Volume constant, then work done is 0. Okay, so now this work done we understood very well. Okay, everything we understood, how it came, how this formula came. dV change in volume, work done increases, change in volume positive, to work done positive. Volume decreases, dV negative, to work done negative. Okay, okay students, so this topic completed. Now one question I will give you. This question you have to solve. This process is given, total 3 processes are given. This is pressure axis, this is volume axis, and all process start from same point A and end at same point B. All process, okay, process A, all process, 3 process are there, start from same point A and they all terminate at point B. Means for all process, initial and final point are common. Okay, first process is like this. This is process 1, A to B. Then second process is like this. Okay. Then third process is like this. Okay. Find the work done order. Find the work done order. First option, W1 equal to W2 equal to W3. Second option, W1 is greater, W2 is greater, W3. W3 is greater, W2 than W1. And fourth option, none of these. Which is correct? Which option is correct? Send your response. A, B, C. Which option is correct? Okay, this is student Navyasri Kotha. Yes, your option correct. Then Bhanu correct. Yashwant correct. Okay, now most of them are sending C, right? Some are sending A, and some students are not sending their response. I'm waiting. You can send your response. Okay, C, yes, correct. So C option is correct. Why C option is correct? What I explained you that if closed process is not given, these all process are not closed. These process are like this, start from A and terminate at B. This is A, this is B. Right or not? So in all these process, what's going on, that these are not a closed process. Only a part is given, A to B, first point. So if such type of process are given, then what we have to do, I explained you that if closed loop is not given, then we discuss here, if closed loop is not given, then join the initial and final point with the volume axis and calculate area. So this concept you have to use here. So what we have to do, this is not closed process. If we talk about first, so first is like this. First is like this, okay. So here in this first process, if we calculate total work done, so what we have to do, join the initial point and final point with the volume axis. This is volume axis, this is pressure. To join initial point, final point, joined. Then calculate area. This is the area. Okay. If we talk about second process, so in second process, this is A, this is B, and this is straight A to B. Okay. Again same point, what you have to do, join the end point with the initial and end point with the volume axis. So this is the area. Clear? And finally that third process is like this, A to B. So for work done, we have to join initial and final point with the volume axis. We joined it. Okay, this much is the area. So you can see in which Out of these three, in which graph area is more? So area is maximum in third, then second, then first. So area is maximum in third graph. So maximum work done. Maximum work done. Is it clear to all? You understood it? Okay. Are work done topic clear to all of you? Send your response. Okay students, so we understood this work done. Now we will move on next topic. So in last class I told you that work done, we will understand work done, then we will talk about internal energy. Okay, so our next topic is internal energy. To understand internal energy, what is internal energy? We will not move so much in the deep. We will only understand the formula and just basic point. What are the basic point here? In last class I explained you that internal energy is nothing but it is simply total energy of the ideal gas. So in mechanics, when we calculate total energy, to what we say? Kinetic energy plus potential energy. Here also kinetic energy plus potential energy. So internal energy of ideal gas is nothing but it is the sum of Ke plus P. P is potential energy. So first point that internal energy of ideal gas, internal energy of an ideal gas is equal to kinetic energy plus potential energy, Ke plus P. Okay, so the energy associated with configuration and random motion of the atoms and molecules within a body is called internal energy. You know, potential energy is energy due to the configuration. So configuration means potential energy, and random motion means kinetic energy, because kinetic energy depends on motion, velocity. Okay, so here this configuration means it is potential energy. They are talking about here potential energy. And random motion means they are talking about here kinetic energy. Is it clear? So random motion K and configuration P of the atoms and molecules within a body is called internal energy. So internal energy we can write that it is the sum of Ke plus P. Now important point, it is the property of the system which depend on the equilibrium state of a system. The internal energy of gas is defined as the potential energy of the molecule, sum of kinetic energy and intermolecular potential energy of molecules. So this one only. Okay. For an ideal gas, the intermolecular forces acting between the molecules is negligible. This is very important point, that intermolecular forces are negligible. This is very important point. What? That intermolecular forces, this point is very important for NEET exam. Intermolecular forces is equal to 0 for ideal gas. Okay. So internal energy, now you have to remember this point, that potential energy is due to intermolecular force. If intermolecular force 0, that's why potential energy is 0. So intermolecular forces are 0, so potential energy of an ideal gas also 0. Okay, so internal energy of an ideal gas is completely equal to kinetic energy because intermolecular force 0. That's why potential energy 0, because potential energy is only due to intermolecular forces. Clear? Okay. So potential energy 0, then we can write that internal energy, generally we represent this internal energy by U. Okay, we represent it by U. This is equal to kinetic energy because potential energy becomes 0. Is it clear? Okay. Now next point. How much is the formula of internal energy? So internal energy equal to kinetic energy, and formula of kinetic energy is F by 2 nRT. This is the formula of internal energy, F by 2 nRT. Now here you have to remember what is F. This F is degree of freedom. This word is new for you. Okay, we will understand it. You have to remember this degree of freedom values for monoatomic gas. For monoatomic gas, degree of freedom is equal to 3. Degree of freedom equal to 3. Why it is 3? What is degree of freedom? We will understand later. For diatomic gas, F equal to 5. So degree of freedom 5 for diatomic gas. Example N2 O2 carbon monoxide. These are the example of diatomic gas. Okay. And for monatomic gas, examples are, helium, neon, argon. All these are monoatomic gases. Clear? So for monoatomic gas, F equal to 3, example helium and neon. For diatomic gas, F equal to 5, example N2O2CO. Other are also there. Formula internal energy F by 2 nRT. What is F? Degree of freedom. N number of moles. R already we understood, gas constant, and T is temperature. Is it clear? Okay, so here N is number of moles, you know it, R gas constant, and T is temperature. Now, if anyone asks, if we change temperature, then internal energy will change or not? Yes, because it directly depends on temperature. So at T2 temperature, it will be F by 2 nR T2, and at T1 temperature, it will be F by 2 nR T1. So change in internal energy, how much? Change in internal energy, how much? F by 2 nR T2 minus F by 2 nR T1. We can take F by 2 nR common, then it become F by 2 nR common T2 minus T1. This is what change in internal energy. If we change temperature, to internal energy also changes. To change in internal energy, how much? F by 2 nR T2 minus T1. What is du? Du is change. Is it clear? Okay. So we understood about internal energy. What is important point? It is equal to K plus P. Potential energy is 0 for ideal gases. Why it is 0? Because intermolecular forces are 0 and potential energy is due to intermolecular forces. So it is 0. So potential energy also 0. So now we can say that internal energy equal to kinetic energy, and kinetic energy formula is F by 2 nRT. This F, degree of freedom, I explained, it is new for you, but remember only this point, that for monatomic gas F equal to 3 and for diatomic gas F equal to 5. Clear? Understood? Okay friends, so we understood today about internal energy also. Now next point, first law of thermodynamics. First law of thermodynamics. I am good, I am fine. Some students are asking me that, sir, how are you? Varsini, okay, she is asking me continuously. So I am good, I am fine, I am very fine. In beginning of class, I told that I am very fine. Okay, and one student is asking, sir, what is capital U? Capital U, array, I represented potential energy by capital U. Okay, this, internal energy. I represented internal energy by capital U. So internal energy is represented by capital U and potential energy is by P. U means internal energy. Okay. Now next, first law of thermodynamics. So we understood everything about work done and internal energy. Okay. So what is first law? The very basic point, first law relate internal energy, work done and heat supplied. Okay, move back on first point. If you remember yesterday when I started the work done concept, how I explained, we are supplying heat to this ideal gas. So what can happen? Ideal gas temperature rise. Temperature rise means internal energy. So whatever heat we are supplying, that heat is distributing, some part of this heat is distributing in internal energy. Okay, well and good. Now next, at same time what will happen? These gas molecule will collide with higher speed with this piston. So there is a chance that this piston can move up, means displacement. Displacement means work done. So whatever energy we are supplying, that energy can contribute in internal energy of the ideal gas, one, and another work done by the ideal gas. This is first law of thermodynamics. This is first law of thermodynamics. Is it clear? What is first law of thermodynamics? Whatever heat we supply, this heat can contribute into the energy, internal energy of ideal gas, otherwise work done. This is only the first law of thermodynamics. Okay, so we are supplying heat, suppose how much? Delta Q. Some part of this heat is distributed in internal energy and some part in work done. So we can write that first law of thermodynamics is equal to heat supplied is equal to some part in work done and some part in internal energy. We are supplying heat, so temperature will change. So we will write here change in internal energy. We are supplying heat, so piston will move. There is a chance, possibility. Means displacement, means change in volume. Change in volume, that is what? Work done. So heat supplied is equal to work done plus change in internal energy. This is what? First law. And very important here, what we are assuming, that total heat is completely distributed in two parts. We are not assuming here any loss in energy. So no loss means energy remain conserved, heat remain conserved. So we can say this law is based on law of conservation of energy. So very basic point, a very important point, that first law of thermodynamics is based on energy conservation. Okay, so you have to remember it. It is based on energy conservation. And how to write it? That heat supplied is equal to work done plus internal change in internal energy. So dq means heat supplied, okay, and dw is work done and du is change in internal energy. We can relate them. We are assuming here no loss. Okay, is it clear? So this is the first law of thermodynamics. Is it clear to everyone? Now here some points are there. When work done will be positive, already we discussed. When work done will be positive, already we discussed. If volume increases, then work done positive, volume decreases, then work done negative. Okay. So about work done we already discussed. When volume will increase, if it is work done by the system or work done by the gas. Okay. So you can read all these points. Further we will understand in next class. Okay students. So till now we understood about internal energy and work done. We done so many questions also today, and finally we relate heat supplied with work done and internal energy. And when work done will be positive negative, we already understood it. About internal energy also we understood, because change in internal energy, we discussed this formula also. It is equal to F by 2 nR T2 minus T1. If temperature increases, then it is positive, because T2 minus T1 will be positive. If temperature decreases, means T2 is less, then it is negative. Is it clear? Okay friends. So yes, you all understood, I think it. Thank you for listening the class. Further we will discuss tomorrow. Okay friends.
Good morning students. So yesterday we have discussed about questions based on calculation of oxidation number, that how to calculate oxidation number. So today we are going to discuss how to relate this with oxidation and reduction. So earlier we have written electronic concept, classical concept, addition of hydrogen, oxygen, these elements. But now on the basis of oxidation number we can say directly. So if we write note point, that increase in oxidation number is called as oxidation. So for any element, if oxidation number increases, then that process will be termed as oxidation. And second point is decrease in oxidation number will be called as reduction. Now we'll only use these points to decide oxidation and reduction. Yes, only these 2 points we'll remember, that increase in oxidation number is called as oxidation and decrease in oxidation number is called as reduction. So now I am going to write some examples. You have to tell that which of the following species is getting oxidized, which of the following is getting reduced by using these method. Increase in oxidation number is called as oxidation and decrease in oxidation number is called as reduction. So let us write first example. I am writing examples. You have to tell which is getting oxidized, which is getting reduced. Mg plus O2 gives MgO. Second example, FeCl2 plus Cl2 gives FeCl3. So what we have to find out that which species is getting oxidized and which species is getting reduced. So try to solve first part. Tell me what is getting oxidized and what is getting reduced. Now both are redox reactions. One species will be oxidized, one species will be reduced. Madhuri is saying magnesium is oxidized. Very good. Magnesium is getting oxidized. Then what is getting reduced in this reaction? Because if it is a redox reaction, then which species will get reduced here? Very good, Madhuri. Oxygen is reduced here. Very good. So what we write here, magnesium oxidation number 0, O2 elemental state 0. Here magnesium is plus 2 and oxygen is minus 2. Now easily we can identify here 0, 2 plus 2. Increase or decrease? Increase. So which process? Oxidation. Magnesium is getting oxidized. Now here O2. 0 to minus 2 oxygen, decrease, so decrease is termed as reduction. So here magnesium is getting oxidized and oxygen is getting reduced. Yeah. Same in second example, you have to tell which species is getting oxidized, which species is getting reduced. FeCl2 plus Cl2 gives FeCl3. Cl2 is reduced. Very good. Fe is oxidized. Very good. Now in FeCl2, ferrous chloride, Fe is plus 2. In Cl2, elemental state 0. Now here FeCl3, iron is plus 3, chloride is minus 1. Now Fe plus 2 to plus 3, increase in oxidation number. So which process this is? Oxidation. Now chlorine 0 to minus 1, decrease, so this is reduction. And so increase oxidation, decrease is reduction. Clear? Now write down next. Now next 2-3 examples I am writing. You have to tell which species is getting oxidized, which species is getting reduced. H2S plus Cl2 gives sulfur plus HCl. Next example is K2MnO4 plus O2 plus H2O gives KMnO4 plus KOH. Next is H2 plus Cl2 gives HCl. So these 3 examples I have written. You have to find out which is getting oxidized, which is getting reduced. So for that you have to write oxidation number for all the species. First, hydrogen is reduced. How it is possible? Plus 1, there is no change in hydrogen. So first write down all the oxidation numbers. Oxidation number in first sulfur, chlorine and then find out which is getting oxidized, reduced. there is no change in hydrogen in first one. Hydrogen is +1 H2S. Here HCl hydrogen is +1. There is no change in oxidation number of hydrogen. Sulfur is reduced. From reactants you have to decide na, H2S, Cl2. What is present here? Sulfur is -2 in H2S. Cl2 0. Here sulfur 0. HCl chlorine is -1. Now H2S contains sulfide ion. So -2 to 0. Increase or decrease? Increase. So this is getting oxidized. Oxidation. So what we say here? Sulfide. Sulfur 2 negative is oxidized to sulfur. And here Cl2 0 to -1. Decrease. So decrease is termed as reduction. So in this case Cl2 is getting reduced and sulfide is getting oxidized. Clear? Now move to next example, second one. And I will highlight. Find out oxidation number for these. In K2MnO4 for manganese, in O2 for oxygen, in KMnO4 for manganese, in KOH for oxygen. Then you can easily identify which will get oxidized, which will get reduced. Try to solve. Magnesium is oxidized. Where is magnesium? There is no magnesium. Manganese is there. So manganese here, if you find +6 oxidation number. Here O2 what will get? 0. Here manganese is +7. Oxygen is -2. Now in manganese +6 to +7. Increase in oxidation number. So this will be termed as oxidation. Now O2 0 to -2. Decrease. So decrease is termed as reduction. So here what we say that manganese is getting oxidized and oxygen is getting reduced. Okay. Now move to next one. H2 plus Cl2 gives HCl. What is getting oxidized? What is getting reduced? Solve next part. H2, Cl2, HCl. H and Cl reduced. How both can be reduced? One will be oxidized, one will be reduced. In H2, what is oxidation number? 0. Cl2 0. HCl hydrogen +1, chlorine -1. Now if we consider this H2 to H positive, it is increase. So increase is called as oxidation. And chlorine 0 to -1, 0 to -1 is decrease. So decrease is termed as reduction. So here hydrogen is getting oxidized and chlorine is getting reduced. Okay. So easily we can find out here that which of the following is a redox reaction. And so this is NEET's favorite question. In NEET there is, you will find many time question this simple, that which of the following is a redox reaction. So this year also in NEET there is a question that which of the following is a redox reaction. So if I give you that question, you have to identify. Now what is question for this year NEET 24? Question is which reaction, not a redox reaction, it is asking which reaction is not a redox reaction. So option first is H2 plus Cl2 gives 2 times of HCl. Option B is barium chloride plus sodium sulfate gives barium sulfate plus sodium chloride, double displacement reaction. Third is Zn plus CuSO4 gives ZnSO4 plus copper, displacement reaction. KClO3 plus I2 gives 2 times of KiO3 plus Cl2. So this is question. What we have to find out that which of the following is not a redox reaction, and this is NEET 2024 and this year question pre. So it is very easy question. So how many students can solve this question? Just you have to tell answer that which of the following is not a redox among these. They are not asking which is redox, they are asking which is not a redox. Which option? First, second, third, fourth. Students are saying first. First we have written right now example. It is example of redox reaction. How it is not redox? This we have written last example, H2 plus Cl2. Now which is non-redox reaction? Second will be the answer. So if we write oxidation number, here hydrogen 0, chlorine 0, hydrogen plus 1, chlorine minus 1. So if you check 0 to plus 1 oxidation, 0 to minus 1 reduction. So reaction in which simultaneous oxidation and reduction takes place, which type of reaction that is? Redox reaction. So first cannot be answer. Now move to next, barium plus 2, and sodium plus 1, chlorine minus 1 and complete SO4 sulfate is minus 2. Here same barium is plus 2, sodium is plus 1, chlorine is minus 1 and complete sulfate is minus 2. So there is no change in oxidation number. Barium remains same, sodium remains same, chlorine remains same. There is no change. So this type of reaction is not a redox reaction. So what will be answer? Second option. If we move to third option, Zn elemental state 0, CuSO4 copper is plus 2, ZnSO4 zinc plus 2 and copper is 0. So if you check here, zinc is getting oxidized, copper is getting reduced. So which type of reaction this is? Redox reaction. And if we move to next, in KClO3, if you find oxidation number of chlorine, you will get plus 5 here. In I2, what oxidation number you will get? 0, elemental state. In KiO3 iodine plus 5 and Cl2 0. So if you check, chlorine is getting reduced, plus 5 to 0, reduction. Iodine is getting oxidized, 0 to plus 5. So this is also which type of reaction? Redox reaction. So they are asking which is not a redox, then what will be answer? Option number 2. So this is a very easy question from this year NEET paper, NEET 2024. So if we can easily find out by oxidation number, if simultaneous oxidation and reduction are present, that is called as redox. If there is no change in oxidation number, that is called as non-redox reaction. Okay. So this is all about oxidation number. Now next term is oxidation state. So if we have to find out oxidation state, what is required? Structure is required. And if we know structure, easily we can find out what is oxidation state of that element. So for that, what is required? A method is required, that is called as charge transfer. What is method? Method is called as charge transfer. So in charge transfer, if there is bond between two atoms, either that bond can be single bond, double bond, triple bond. I can't find oxidation number, please explain. For that you have to listen last 2 classes. You are coming in between the class and you are saying explain complete oxidation number. Not possible now. Now first point is, if there are 2 atoms and what we are assuming that electronegativity of A is more than electronegativity of B. Now En means electronegativity, just we are assuming that. Electronegativity. So in periodic table, when we are moving left to right, But we are moving left to right in periodic table, then electronegativity increases. This is general trend. Just remember this. Whenever we are moving left to right in a period, respective period, electronegativity will increase. And if you move down the group, when you move down the group in periodic table, what will happen? Electronegativity will decrease. So this is general trend, that left to right increase, down the group it will decrease. So easily we can identify that which element is more electronegative, which element is less electronegative. Okay. Now after finding this electronegativity, three cases we have to remember. If between A and B single bond is present, so if we break this bond, electronegative element will get negative charge, less electronegative will get positive charge. So what charge we write? A will get -1, B will get +1. So -1 +1. Clear? Second is, if there is double bond between A and B. So for A, what we write? -2. For B, what we write? +2. More electronegative will get negative charge. So in double bond, what charge we have to write? +2 and -2. If there is triple bond between two atoms, then what will be charge here? -3 and +3. -3 means triple bond means more electronegative getting -3, less electronegative getting +3. So for single bond, charge separation +1 -1. For double bond, +2 -2. And for triple bond, +3 and -3. And if there are same atoms, A, no difference in electronegativity. So if there is no difference in electronegativity, then what we have to write? 0, 0. Means there is no charge separation. So what we have to remember here? Single bond +1 -1, double bond +2 -2, triple bond +3 -3. And if there is no difference in electronegativity between two atoms, then 0 and 0. So by this we can easily find oxidation state. So let us take first example, HCN molecule. So in HCN, what is structure? Hydrogen, carbon, triple bond, nitrogen. What is structure? Hydrogen, carbon, triple bond, nitrogen. Now first we have to break this bond, carbon and hydrogen. Which is more electronegative? Carbon. Which is less electronegative? Hydrogen. So hydrogen will get +1, carbon will get -1. So single bond we have broken. Now break this triple bond, carbon, nitrogen. Which is more electronegative? Nitrogen is more electronegative than carbon. So this will get -3, this will get +3. So if I write what is total oxidation number or oxidation state of carbon here? +3 -1, what is left? +2. What is oxidation state of nitrogen here? -3. What is oxidation state of hydrogen here? +1. Reason is because if I write electronegativity order here, which is most electronegative? Nitrogen, then carbon, and least is hydrogen. So first you have to break single bond, +1 -1. Then break this triple bond, +3 -3. So +3 -1, what is net charge left? +2. So oxidation state of carbon in this compound is +2. Oxidation state of nitrogen in this compound is -3. And what is oxidation state of hydrogen? +1. So break these bond. First we have to break single bond +1 -1. Then we are breaking triple bond +3 -3. Which is more electronegative? Nitrogen. Next is carbon. Least electronegative is hydrogen. Check this example. Any doubt in this? Any doubt in this HCN? Yeah. Now next I will give one example. You have to tell me oxidation number for sulfur in that compound. So next example is, but I will write structure for you. H2S2O8. Common name for this compound, it is called as Marshall's acid. So I am writing structure for you. Then you have to tell me oxidation number of sulfur. OH. So 2 sulfur are present. For these 2 sulfur, what we have to find? Oxidation number. Now take your time, try to solve. There is no hurry. Solve very slowly. Just break all the bonds of both sulfur and try to find what is oxidation number here. Madhuri, you are not breaking all the bonds. You are calculating like this: 2 hydrogen, 2 sulfur, 8 oxygen. You have to break bonds and then find out oxidation state. For sulfur, +7 is never possible. Maximum state of sulfur is +6. Sulfur can never show +7 oxidation state. Maximum is +7. Oxidation number state both will be same here. That's why I am saying oxidation number, because you will get incorrect answer. You will write here 7. I am asking oxidation number. For that, what you have to find first? Oxidation state. Average of oxidation state is oxidation number. +6. Many students are saying +6. Very good. So let us start with this sulfur. First I am writing which is more electronegative. So if I write electronegativity, oxygen is more electronegative than sulfur, and oxygen is more electronegative than hydrogen also. So if I write first sulfur double bond oxygen will get -2, sulfur will get +2. Clear? Now here double bond again -2, this will get +2. Single bond, this oxygen will get -1. So sulfur will get +1. Here also single bond -1, sulfur will get +1. So all the bonds we have broken, but we are getting here +6. But we are getting here +6. Now same we have to write similar for this sulfur. -2 oxygen, sulfur will get +2. -2 oxygen, sulfur will get +2. Single bond -1. Here what we get? +1. Here -1, but we get +1. So what we are getting here? +6. So both sulfur are present in which oxidation state? +6. So if I write oxidation state of sulfur, so what is oxidation state? +6 and +6. And what is oxidation number of sulfur? Oxidation number is average. So +6 divided by 2. Again, what I am getting here? +6. Now here oxidation number and oxidation state both are same. But why we are getting here +7? If you apply this method, 2 times of hydrogen +1, 2 times of X, 8 into -2 is equal to 0. So what do you get here? +7, but this +7 is not possible. Now why, Madhuri, you are getting incorrect this? We are writing +7. Because what we are assumed here that all 8 oxygen are present in which oxidation number? -2. But here if you check, 2 oxygen are present in which oxidation number? -1. Why we are getting by this method incorrect? We are getting +7. What is reason for that? Because in this method we have assumed that all 8 oxygen are present in which oxidation number? -2. But here if you check, 2 oxygen atoms are present in which oxidation number? -1. That is called as peroxide linkage. That is called as which type of linkage? Peroxide linkage. Okay. So what is important point about peroxide linkage? So what extra point I can write? If it is A, oxygen, B, this linkage is called as peroxide or peroxy linkage. And whenever this type of linkage is present, oxygen has which oxidation number here? -1. So that's why we are getting there +7. Now check, students who are getting +7, now they can understand that why oxidation number is not +7 and it is +6. Because all sulfur are not in, all oxygen are not in -2. 2 oxygens are present in which oxidation number? -1. Check this. Any doubt? Check this example. Marshall's acid. How to find oxidation state? Ratna, you answered correct, +6. There is one. Madhuri, now clear? Okay. Now let us move to next one. So in earlier classes, when we are writing difference in oxidation number and oxidation state, I have taken one example. H2S4O6, where I am directly writing that 2 sulfur are present in different, 2 sulfur are present in different. So in this structure, you have to find what is oxidation state of sulfur. Structure I am writing. But you have to find oxidation state of sulfur. Try to find. For 4 sulfurs are present. For every individual sulfur, you have to tell oxidation state. +5. Ratna, correct. +5, 0, 0. Madhuri, again you are getting +7. Structure is given. How you are getting +7? Now in this case, you never get +7. If you apply that X method also, old method, then also will not get +7. So many students, because earlier we have written this 2 sulfur +5, 2 sulfur 0. Now method we should know. So what method we have to apply? So which is more electronegative? We know oxygen, -2 for sulfur, +2. -2, sulfur will get +2. Now both are sulfur. So there is no charge separation, 0 and 0. And -1, +1. So what we get here? +4, +1. Total is +5. But we are getting here +5 here. Now if we write for here -2, +2, -2, +2. Oxygen, single bond -1, +1. Here both sulfur 0. This is also 0. So what we get here? +5. So if you check here, how many are present? 4 sulfur are present. 2 sulfur are in +5 and 2 sulfur are in 0. So if they ask what is oxidation state of sulfur, so oxidation state of sulfur is +5, 0, 0 and +5. And if they ask what is average, so average is +5, 0, 0, +5 divided by 4, what you get? +5/2. 10/4, that is 2.5. Here. So if they ask that what is average of oxidation state, then answer is 5/2. If they ask what is individual oxidation state of sulfur, 2 sulfur has +5 oxidation state, 2 sulfur has 0 oxidation state. So what will be answer? +5, 0, 0 and +5. Any doubt in this example? Now it is clear why we have written at that time 2 sulfur +5, 2 sulfur 0. We can find easily. Let us write next example. Next question is H2SO5. Common name for this acid, it is called as Carrow's acid. So sulfur, I am writing structure for you. OH, oxygen, oxygen, hydrogen. Now only one sulfur is present, so number and state is same. Try to find out what is oxidation number of sulfur in this compound. H2SO5. Madhuri, again you are writing incorrect. Try to solve again. It is not 4. All other students are telling correct. 6 they are saying. That is correct. 4 is not possible here. If you take this sulfur, double bond -2 +2. Again here double bond -2 +2. Here oxygen single bond -1 +1. Again single bond -1 +1. So what we get here? +6. So what is oxidation state of sulfur here? It is +6. Now remember, this oxygen is also in -1. So which type of linkage is present here? This linkage is peroxy linkage. So which type of acid it is? A peroxy acid. It contains oxygen-oxygen linkage. Clear? So remember this, in H2SO5 also what is present? Peroxy linkage, and oxidation state of sulfur will be +6. Any doubt in this? Clear? Now let us take next example. Next example is H3 PO5, phosphorus, double bond oxygen, OH, OH, oxygen, oxygen, hydrogen. So for phosphorus, what we have to tell, what is oxidation state? Try to solve. So maximum students are writing answer +5. Very good. Answer is +5. Very good. So what we write here? Oxygen is -2 because double bond is present. +2. Here what is present? Single bond -1 +1. Single bond -1 +1. Here also -1 +1. So what we get here? +5. And here this oxygen is also -1. So what is oxidation state of phosphorus here? +5. And this linkage is also oxygen-oxygen linkage is called as which linkage? Peroxy linkage. So when you calculate by this, for example, if you take this method, 3 hydrogen, 3 into +1 plus X, 5 into -2 is equal to 0. What you get here? 10, 3 plus 7. But this is also not possible, because maximum oxidation state of phosphorus is +5. It can never show +7. Why we are getting from this incorrect? Because we have assumed that all 5 oxygen are in which state? -2. But here which linkage is present? Peroxy linkage. That's why answer is +5. Clear? So whenever these type of peroxy acids are present, we'll draw the structure and then find out oxidation state. So is it clear to you how to find oxidation state? So in these examples, we are not taking any case where difference of hybridization is there, because we have not studied about hybridization till now in chemical bonding. So when you got about that hybridization, then you can do that. If two atoms have different hybridizations, then what will happen? Their electronegativity will be different, then there will be charge separation also. So those examples we are not taking. We have taken simple examples here where there is no difference in hybridization. If same type of atoms are there, they have same hybridization. Clear? So this is all about oxidation number and oxidation state. Can we count the bond between sulfur and sulfur? No. No need of that. For structure, if we know, then we can find. So in next class, you will get to know about balancing of redox reactions and all these terms. So students, thank you for today. This is all about oxidation number and oxidation state.
Good morning, students. Good morning. Good morning, all of you. Good morning, Kiran. Good morning. Good morning. Yes, good morning. Yeah, I'm good, Sindhu. Sanjana, yes, good morning. Yes, all. Good morning, good morning. Okay. Okay. I'm good, Sirisha. Yeah, Deeptika, I'm good, I'm good. Good morning, good morning. Yeah, Neer, I'm good. Afifa, I'm good. Okay. Madhuri, okay, good morning, good morning. Finally, we have come to the end of this chapter. King of, what is that? King of the Arun. Okay. I'm good, Amma. So we have come to the end of this biological classification chapter, because you have learnt already the first part, starting with Monera, then Protista, then fungi. Now today we'll be seeing that remaining part of the RH Whittaker 5 kingdom system of classification. Yes, Samreen, all of you. Yeah. Okay, good morning. Good morning, Anunaya. Good morning. So now I'll ask quick questions and later we'll proceed to the today's topic. Okay. So yesterday I have completed the fungi, right? So study of fungi is called as, yeah, answer. Yeah, Lekhya, good morning. Yeah. Samiksha, okay. Mycology, yes, Anunaya, mycology, very good. Samreen, yes, mycology, yes, very good. Good, mycology. Yes. Now, what is the body of the fungus called as? Body. The structure or the body of the fungus is called as? Body of the fungus, it is called as. Very good, Anunaya, mycelium. Kiran Kumar, very good. Very good, Samreen, yes. All subject corner, yes, very good. Mycelium, very good. One filament in the mycelium is called as, if it is looking, okay. Yeah, now it has come. Yeah, hyphae. Network of hyphae will form the mycelium. Okay. Good. What are the cross walls in the fungal filaments called as? Cross walls in the fungal filaments. What are these called as? Cross walls in the fungal filaments are called as? Yes. Sorry. Yes. Septa. Correct. Septum singular, septa plural. Very good. What is this condition called as? All of you can see on the board. This condition in the fungal filaments is called as? Can see this? Very good, Samrin. Cenocytic. Very good. Aseptate. Very good. This is called as aseptate and cenocytic condition. Cenocytic condition. What are the steps of sexual reproduction? Good. Multinucleate, cenocytic, aseptate. Steps of sexual reproduction. Phycomystis is a class name, Afifa. Yeah. 3 steps. What are those 3 in the sequence? Yeah, honey. First one, yes, plasmogamy. Next, second one, karyogamy. Very good, honey. Yes, Samreen, plasmogamy, karyogamy, zygotic meiosis. Very good, very good. Sanjana, yes, very good. So the sequence is first plasmogamy, then karyogamy, and final step is zygotic meiosis. Good. Okay, fungi will show spores in two reproductions. In which reproduction fungi will show spores? Vegetative, asexual, sexual. Among these three, which ones will show spores? Honey, only asexual? Sairaj, sexual. Okay. Naveed, vegetative? No. In the vegetative reproduction, there are no spores. Both in asexual reproduction and sexual reproduction, they will show the spores. Asexual spores are there, sexual spores are there. Okay, asexual and sexual, yes, asexual and sexual. Last question. Name the only one motile spores of fungi. Only one motile spores of fungi. Motile spores, very good, Afifa. Yes. Whose spores are not motile, ma? Whose spores are non-motile? All subject corner, yes, juice spores, correct. Samyan, yeah, juice spores, correct. Yes, juice spores are the motile spores. Whose spores non-motile? Jew spores are motile, Jew. These are the motile spores. Okay. So that's all for now. You will be learning after coming here based on the nature of the mycelium, mode of spore formation, okay, and based on the septa. They have been divided into 4 classes. Fungi have been divided into 4 classes. That you will be learning after coming here. Classification, okay. Now let us see today's topic. After that you have plantae and animals, animal kingdom. Because we are done with the 3 kingdoms, Monera, Protista, fungi. The last 2 kingdoms are left out, that is plants and animals, plantae and animalia. Plants are chlorophyll containing, they contain chlorophyll. Chlorophyll is present here, in animals chlorophyll is not there. These are generally autotrophic, autotrophs, but these are heterotrophs. They contain cell wall. Cell wall is absent. Here cell wall is made up of cellulose. Okay. And the cell wall is absent, so no cell wall materials will be there. These have complex body organization, complex bodies, because they have the sensory system, neural system, okay, then locomotory system, because most of them will show the locomotion. They have pattern formation, growth, okay, they have holozoic mode of nutrition, holozoic mode of nutrition, okay, these all things will be there in the animals. Whereas here they are autotrophs, but some are partial heterotrophs are also present, partial heterotrophs, like insectivorous plants. Insectivorous plants, because they eat the insects. Insectivorous plants. And even parasitic plants also we have. Parasitic plants. They will come under partial heterotrophs. These are the basic differences between plantae and animalia. So we have covered with the 5 kingdoms. About these, about plants you will be learning in the plant kingdom, all the remaining details. About animals, in the animal kingdom of zoology, you will be learning other details. Just the basic information about the plants and animals. Now, let us see the next one. Viruses. Okay, viroids, prions, and lichens. Did you come across any of these in the R.H. Whittaker 5 kingdom system of classification? Did you come across any of these in the R.H. Whittaker 5 kingdom classification as examples? Yeah, plants are classified into algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Yeah, that you will be learning later in the plant kingdom. Okay, no, yes. You did not get any of these 4 in the R.H. Whittaker 5 kingdom classification. So these organisms did not find place in 5 kingdom system of classification. That you are going to learn today. Now, viruses. First one, viruses. Viruses are made up of nucleic acids and proteins. So nucleoproteinaceous particles. They are made up of nucleic acids and proteins. So nucleoproteins, but these are lacking. They lack cytoplasm, they lack the nucleus, they lack the membrane bound cell organelles. Nothing is there. No cytoplasm, no nucleus, no membrane bound organelles, no cell wall, no plasma membrane. So can you consider this as a living or non-living? Because it shows both the properties of living and non-living. These are called connecting link between living and non-living organisms. Living and non-living. Because it shows both the properties. Viruses will show both the properties of living as well as non-living. You know, like non-living things, they will be staying outside the host, like crystal. Okay, you can see the crystal form of the virus. So they'll be non-living and they'll be like crystallized forms are there, and they won't do anything. They are inert outside the cell. They remain inert outside the cell. That is a non-living property. Living property, when you keep that virus, when the virus enters into the cell, inside the cell it multiplies or replicates. That is a living process. So living property. So multiplication inside the host cell or replicating inside the host cell will make the virus living. So as it is showing both, depends on whether it is present inside the host or outside the host. It is called connecting link between living and non-living. But as I said, they don't have any cells. Cytoplasm, cell wall, plasma membrane are not there. These viruses are called as non-cellular or acellular. Or you can call them as acellular. They are called as non-cellular or acellular. But for their multiplication, definitely they need a host. So they are called as obligate intracellular parasites. Obligate intracellular parasites. Obligate means compulsory, strict. They need, for their multiplication, they always lives inside the cell of a host. They are living inside the host cells particularly. They need for their multiplication the host. So they are called obligate intracellular parasites. And these viruses, they are made up of nucleoproteins. But first time, who discovered that virus and how they were, their functions are known, or what they are doing, what their diseases are caused by them, or whether any virus is helpful for us, all those things started with the experiments. So one scientist is there, Dmitry Ivanovsky. Dmitry Ivanovsky first observed that these are the smaller ones, okay, and they can be passed through the bacterial filters. They can pass through the bacterial filters, which are actually trapping bacteria. These ones can pass through the bacterial filters. That means these are smaller than the bacteria. Okay, these are identified by Ivanovsky for the first time. Small particles, smaller than bacteria, they can pass through the bacterial filters. So Ivanovsky first observed that viruses, that they can pass through the bacterial filters, and they caused the disease called as tobacco mosaic disease. That is a causative agent of tobacco mosaic disease. Later, okay, first discovered by Ivanovsky, and later the scientists came to know about the tobacco mosaic disease is caused by the virus. Some virus is there that will cause that. This identification was done by M.W. Beijering. Beijering identified that the causative agent of the tobacco mosaic disease is a virus. Okay, how he identified means he took the infected plant and he added the infected plant extract to the healthy plant. That also had shown the same symptoms. So he identified that the disease is caused by a particular virus named as TMV, tobacco mosaic virus. Okay, but later that infectious fluid was called by Beijering as contagium vivum fluidum. Contagium vivum fluidum, nothing but infectious fluid. Okay, and he called this infectious fluid like that, and he gave the name virus for that. Name virus was given by Beijering. Okay, that is about these two scientists. Then next one, W.M. Stanley crystallized the virus. First time crystallized the virus, W.M. Stanley. He crystallized the virus for the first time. So Ivanovsky was the first person to identify the virus that they can pass through the bacterial filters. They are the smaller than bacteria. He discovered first viruses basically, Ivanovsky. Then Beijering called that infectious fluid as contagium vivum fluidum, and he named it as virus that will cause the tobacco mosaic disease. W.M. Stanley crystallized the virus for the first time, which can be inert outside the host. We can take the crystals, they will have the virus and nothing it will do. These are the works of these three scientists. Now let us see the virus structure and examples. Before going to that, I would like to tell you they are made up of two things, nucleic acid and proteins. Okay, nucleoproteins already I have given you. So nucleoproteins means, this means nucleic acids. Nucleic acids we have either DNA or RNA. Anything can be there in the nucleic acid component in the structure of the virus. Then this protein coat, protein will be forming a coat which is called as capsid. Okay, different types of proteins will be there, and that proteins in the capsid are called as capsomeres. Capsomeres. So generally the viruses are made up of nucleic acids and proteins, nucleoproteinaceous particles. That nucleic acid can be either DNA or RNA. If you take plant virus, plant viruses generally they'll have single stranded RNA. Generally, animal viruses will have either single stranded RNA, okay, or double stranded or single stranded DNA. But remember, never both are present. Never both are present. No virus, not even a single virus is having both of them together. Either this or this will be there. In the center, that is a nucleic acid, which is causing that multiplication. This is the genetic material. This is infectious. Nucleic acids are infectious because they will form the new virus particles. So infectious component among these two is the nucleic acids, not the proteins. Proteins are not infectious, they cannot cause any viral infections. Only the nucleic acids can cause the infections. Plant viruses generally they have the single stranded RNA as the genetic material, but animal viruses they can have anything. They can have single stranded RNA, double stranded DNA, single stranded DNA, anything, any one, never both. Any one will be there. Okay, have all understood up to here? DNA or RNA, yes, DNA or RNA, never both together. Virus contains both RNA and DNA. No, it will not contain both. It will contain either, any one, either DNA or RNA, never both. Ma'am, okay, leucovirus has both RNA and DNA, no ma'am. See ma, you have to follow NCERT. Okay, in NCERT all of you after buying the NCERT. If you have NCERT, you can see that. Okay. Don't go beyond NCERT. Macrophagy. Oh, yeah, got it. Leucovirus is not mentioned in your syllabus. You are going to learn your syllabus from NCERT, then only you can crack the NEET. And by the way, I would like to tell you, NCERT is not updated since so many years. So updated ones you will not get. Okay. They asked about leucovirus. Where, ma? Ramnath, where, when they asked leucovirus? When they asked? I haven't seen so far. Leucovirus. Yeah, any further doubts? Understood? What are capsomeres? Yeah, I'll show you. I'll show you now. Virus can show both characters, yes. Living as well as non-living characters. Living characters will be shown when it is inside the host. When you keep it on the bench or somewhere, it is non-living. It shows both the properties. Yeah, okay. Now let us see the next one about the structure. All of you can see here. This is the structure of a plant virus which is given in your syllabus, that is TMV. T stands for tobacco, M stands for mosaic, V stands for virus, tobacco mosaic virus. It has, any virus should have two components, one is nucleic acid, another one is proteins, protein coat. So you can see here, this green color balls are there, that will be running in the center like this. Continuation. Okay, if you want, I'll show you the same color. But we can't see the inside one because it is, these proteins are around that. Okay, so this is the single stranded RNA. RNA, single stranded RNA will be there in the center. And that single stranded RNA will be covered on all the sides by the capsomeres. These are nothing but each one, you can see each one, single one, they are called as capsomeres. They are nothing but protein subunits, protein units or subunits. So protein units which are forming the coat, protein coat or capsid, they are called as capsomeres. Someone asked about capsomeres. What are capsomeres? They are just protein units which will form a protein coat. That protein coat is called as capsid. What is the name of the protein coat in the virus called as? Capsid. So externally, for providing the nucleic acid protection, externally, nucleic acid will be there in the center. And around that nucleic acid in a helical manner, helical symmetry, all these protein units will be there in the helical symmetry, which gives protection to the inner nucleic acid. And that protein units will be called as capsomeres, and the total protein unit cover is called as capsid. Okay. So capsid is made up of capsomeres, they are the protein units, protective coat. This is giving protection to the internally present nucleic acid. In TMV, we have single stranded RNA. As mentioned in NCERT, most of the plant viruses will be having single stranded RNA. The common genetic material of plant viruses is single stranded RNA. Okay. This is the structure of TMV. Have all understood up to here? Capsid is made up of capsomeres arranged in polyhedral, yes, consists of geometry. Yeah. See, this shows helical symmetry. Helically it is present. TMV shows helical symmetry. That means that capsomeres will be arranged like a helix form. Like this, the capsomeres are arranged, helical form around that nucleic acid. But if it is showing the polyhedral symmetry or icosahedron symmetry, it will be having a different shape. Okay. This is the causative agent of tobacco mosaic disease. Okay. Now the next one, these are bacteriophages. This is called bacteriophage because it will infect bacteria. Phage means it is a virus which infects the bacteria. It is called bacteriophage. A typical bacteriophage also has nucleic acid and protein coat only. So the total pink one, whatever you are seeing, the pink one, everything is capsid. It is made up of different capsomeres, protein units. And in that protein unit, that capsid center, nucleic acid is present. This yellow one, what you see here, yellow one, this is the DNA. The nucleic acid is being protected by the capsid. And bacteriophages will be having generally double-stranded DNA. This genetic material is double-stranded DNA. Generally, the bacteriophages, most of the bacteriophages will have the double-stranded DNA in the center, surrounded by the capsomeres. So, this will be looking like a tadpole shape. This will be having the head portion. This is called as head. This is called as tail. So, it looks like tadpole, a larva of frog, tadpole shape. This will be, the head will be showing the polyhedral symmetry. Polyhedral will be looking like this, or icosahedron symmetry will be looking roughly like this. This is somewhat the polyhedral symmetry or icosahedron symmetry. So, this kind of symmetry will be shown by the head portion, but tail will be showing the helical symmetry. So, it will be showing both, here polyhedral symmetry, here helical symmetry. So, it shows binal symmetry. Tail and head are connected by something called as collar in between, and tail will be providing with the spikes, tail fibers with the base plate, and tail is covered by a contractile sheath. Okay, so this is the basic structure of the bacteriophage. The remaining details after coming here you will be learning. Just nucleic acid and protein coat will be there. And the protein coat is made up of capsomeres. This is the shape and structure of the bacteriophage which infects the bacteria. Yeah. Any doubts more in this? Yeah, understood? At present in the market, can we get updated NCERT? You will get 2023 you have to buy. Okay, understood everything? Yes. Okay, good. So, we have finished with the virus. Now, can you tell me any viral diseases which infect humans? Viral diseases. What viral diseases will get? Is bacteriophage harmful to humans? No, it's not. It will only kill the bacteria. Okay, bacteriophage cause mumps and smallpox? No. Smallpox and mumps are human diseases. Yeah, COVID-19, very good. Why you are not taking the common cold? Yes, cold, very good. Someone told cold, smallpox, mumps, yes. What about AIDS? HIV is a virus name. AIDS is a disease. Yes, AIDS. TB is not viral. TB, tuberculosis, is caused by bacterium. Don't say TB. Herpes, okay. Smallpox, chickenpox, these are all viral. Dengue fever, yes. Dengue virus, influenza virus, yes. Flu virus, yes. Okay. Got it? Yes. Coughing, that may be because of cold infection or different types of coughs are there. TB also can cause cough. Ebola virus, very good. Ebola. Yes, Ebola disease, okay. Malaria is caused by protozoan parasite, Plasmodium, not viral. Polio, yes, viral. Smallpox, viral. Yeah, Ebola. HIV is a virus name, don't say HIV. AIDS. disease names. Yeah. Plant disease names, already you have got one plant virus, tobacco mosaic virus. So, it causes tobacco mosaic disease. One example is enough. Remaining diseases, plant viral diseases, you will be learning after coming here, like rolling, curling, okay. So many other diseases are there by the virus in the plants. Chikungunya, yes, virus. So, with that we have completed the virus. The next one, you can see here, we have completed with the virus. Inside this we have the nucleic acid and this is the capsid. Capsid is the outermost protein coat which covers the nucleic acid. Then viroids. Can you see here something it is given in the center? This is the actual photograph which we have taken from the net. This is the viroid. It is a single stranded RNA. Single stranded RNA without any protein coat, and it has low molecular weight. So, viroid is not having the protein coat. It is made up of only nucleic acids. Only it is made up of single stranded RNA, and that RNA also has less molecular weight. No protein coats. These viroids will cause diseases in the plants particularly. It causes the disease called potato spindle tuber disease. Potato spindle tuber disease. They can cause diseases only in the plants, okay. Citrus exocortosis viroid, like that. Plant diseases are caused by this, and this viroids will have only nucleic acid. There is no protein coat, no capsid, and always they will be having single stranded RNA with low molecular weight, and they cause the disease called potato spindle tuber disease. Okay. Next one, you can see here prion. Prion. These are almost equal to the size of the virus, equal to the size. The size of the virus and prion will be almost equal, but prions, they are made up of only proteins, no nucleic acids. Only proteins. Infectious proteinaceous particles are called prions. They don't have nucleic acid. They are made up of only proteins, and they will cause generally neurological disorders. Neurological diseases are caused by the prions. These are abnormally folded proteins. Abnormally folded proteins, and they are infectious protein molecules, and they will be causing neurological diseases. Okay. These prions will be causing diseases in the animals. Animals only. Animals. The examples of these prion diseases are bovine spongiform encephalopathy and CJD. These two are equal. BSE is there in the cattle. Bovine spongiform encephalopathy is a cattle disease which is caused by prions. Then CJD, Creutzfeldt-Jakob disease, that is caused in humans by eating the infected cattle meat. Infected cattle meat if we consume, then humans will get that Creutzfeldt-Jakob disease. The people will have lesions in their brain, nervous system, and they'll have the madness. So, in the cattle, that BSE is also called as BSE is also called as mad cow disease, because the cow will be behaving like mad. The expansion for this, bovine spongiform encephalopathy. Okay, this is affecting the cattle. CJD, Creutzfeldt-Jakob disease. Both are neurological. Neurological diseases caused by prions. Infectious protein particles are called as prions. They will cause these kind of diseases. Okay. How all understood, ma? Yeah, any doubts you can ask now. Any doubts? Yeah, Sanjana, prions, infectious proteinaceous particles. No doubts. If any doubts are there so far, you can ask now. Understood? Dheeraj, what is your doubt? How it get infected? What get infected? Does prions contain capsid? No. Prions are made up of only proteins, amma. Viroids are made up of only nucleic acids. Viruses are made up of both proteins and nucleic acids. Yes, classes will be going on here also, so don't worry. We'll repeat here when the actual topic comes. Sharmila, Diner, yeah, you will be learning about extra also, scientist names and all. Yes, Diner, Prusiner. Okay, don't worry. Whatever I taught, that much is enough now. Yeah. BSc is curable? No. Neurological, you can't remove the proteins clearly, but we have treatment, not completely curable. Not completely curable, Sairaj. Will you tell plant kingdom tomorrow? Yeah, in the next class, they will. It will start with the plant kingdom, so don't worry. Just 4 brief points I have told you. They contain cell wall. Cell wall is made up of cellulose. They contain chlorophyll. They are multicellular eukaryotes and generally autotrophs. They can perform photosynthesis. Difference between DNA and RNA, it has been done already in biomolecules. Deoxyribonucleic acid and ribonucleic acid. Anju. What is that? Okay. Next. Any covering layer around prions? No, no coverings. Even for viroid and prions, there are no coverings. BSE is only caused in cows, ma'am. It can be caused like in any of the cattle. Generally, mad cow disease. Cows are more affected. Any cattle. Viroids and prions are discovered by whom? Okay, Diner and Prusiner. Viroids are discovered by Diner, prions are discovered by Prusiner. Or these diseases could be cured? Not permanently, yes, treatment is available. Then what about what is that? Basidiocytes? Okay, done. Viroid affects humans or animals. Viroid will affect only plants. Sanjana, only plants. Prions will affect only animals. Viruses can affect both plants, animals, humans. Viroids only plants. Cause diseases in humans? No, viroids will not affect humans, animals. How is mad cow disease spreads? By eating the cow meat, which is having that infection, disease. Yes, you should buy NCRT. Jyotika, I am good. How are viroids discovered? When they were doing some experiment, they discovered randomly. Okay? Okay. Anju. Then, done, understood? Yeah, beef. Yes, beef only, correct. Infected beef, not normal beef. Yes, done. Now, Sanjana, let us see the lichens. the last one. So all of you can see here, these are the lichens. Okay, this total green one which is there on the bark of a tree, this one, this is white. So this will be the foliose one. It will be looking like this. This is the bark of a tree. Here you can see the boundary. This is the bark on a tree bark. Okay, it is present. And these lichens are the symbiotic association between one alga and one fungus. So lichens are made up of an alga and a fungus. Okay, that algal component is called as phycobiont. Fungal component or partner is called as mycobiont. Okay, two partners will be there. They are having the symbiotic associationship. Algal partner is called as phycobiont and fungal partner is called as mycobiont. You already know term mycology, study of fungi. So fungal partner is called as mycobiont and algal partner is called phycobiont. And these two will have a symbiotic association, one alga and one fungus. The alga is called as phycobiont. This fungus is called mycobiont. They live together in a symbiotic association. That symbiotic association is called as lichens. These are eukaryotic. Unlike that viruses, viroids and prions, okay, these are eukaryotic organisms because it is a combination of two organisms which are eukaryotes. And this alga will be giving food for the fungus. In return, the fungus will be giving shelter to the alga plus water and minerals are provided by the fungus to the alga. So alga will perform the photosynthesis because they contain chlorophyll and it will form the food. That food will be provided to the fungus. Fungus in return, it will be giving shelter. It will be keeping the alga in its structure. Shelter it will provide, water and minerals will be provided by the fungus to the alga. So symbiotic associationship, mutual beneficial associationship. One is giving something, other one is also giving something. Symbiotic association is called as lichens. And these lichens, generally they are present on the trees and different forms of lichens are there. And these lichens are very good pollution indicators. They will never grow in the polluted areas. Okay. Lichens are good pollution indicators. Pollution indicators. That means you don't think that if the lichen is there, it will indicate the pollution. No. If lichens are not there, they will indicate that area is polluted. Okay. They grow in non-polluted areas. Non-polluted areas. They cannot grow in the polluted areas. Particularly sulfur dioxide pollution will be easily identified by lichens. Lichens cannot grow in the sulfur dioxide polluted areas. Any other, but they will never grow in the polluted areas. So in that way, they are indicating us pollution whether it is there or not. If pollution is there, they will not grow. If pollution is not there in that area, then you can see the lichens. So in your area, if you see the lichens, then you can think that your area is unpolluted or non-polluted. If you can't see any lichens, that shows that you have in your area air pollution, particularly air pollution. I hope all of you understood. Any doubts in this? Any doubts? You can ask me now. Can lichens present on animals? No, on animals, ma. Not on animals. Generally on plants. I mean, the basic surface, soil, rocks, like that. No doubts. Okay. Okay, Anunaya. No doubts, Lekya. Good. Are they living? Yes, they are living. Lichens, they are eukaryotic. Alga and fungi are living, no? Lichens is not a plant. It is an association of alga and fungus. Alga and fungus. They are eukaryotic, living, cellular, all multicellular. Many cells are there. Okay. Do they cause any disease? No disease causing them. They are useful for us generally, lichens. Lichens do not cause any disease. They are useful. They are used as spices also in the biryani and all. Okay. Is lichen a flowering plant? It is not a plant at all. No flowers, no plant. It's not a plant. Yeah, litmus paper production. Yes, correct. Lichens are used. Yeah, spices. Yes. That's all. Okay, if you learn properly, then you are going to find it easy. And if you listen to the classes also, you will find it easy. Okay, I hope all of you understood. Okay, you can continue with your next classes. Thank you. Thank you so much.
Good morning, students. Hello. Good morning, students. How are you? Yeah, very good morning to all of you. Yes. How are you? I'm good. Fine. Thank you. Yes, doing good. I'm doing good. Yes, Ratna Buji, I'm very good. Fine. Vemanarayana Jojari. Yes, people are here to listen to class. What was that question? Yes, I'm listening to you. Yes, I'm good. Yeah, okay. So coming back, as I mentioned you earlier also, human body was one such very complicated machine which was not discovered completely till now. Even though we got information regarding many types of body systems, we have got integumentary, circulatory, respiratory, digestive, excretory, muscular, skeletal, neural, sensory, endocrine, reproductive systems and immune system also, but doesn't put a full stop there. No, there are few more things which were on their way of discovering, and might be you guys might be taking that chance to discover new things about human body. So today, let us assume human body as an engine and talk about that. So in a bike or car engine, if you give it a fuel and oxygen, definitely oxygen will be used as an ignition. We want that for igniting fuel, right? So without oxygen, fuel cannot be oxidized properly. Similarly, without oxygen, food molecules cannot be oxidized properly, isn't it? So question to you, does every vehicle get filled in with same fuel? No. Fuel might be petrol, diesel, sometimes kerosene, CNG, biogas. There are many types of fuels and every fuel does not have exactly same composition. They may differ in their components. Therefore, whenever any sort of fuel gets combusted, the byproducts, pollution which they give out is not exactly same. It may differ. Petrol may give you less smoke, but kerosene may give you more smoke because of more particulate content in kerosene, right? Similarly, you eat carbs, proteins, lipids. We digest them properly. We try to extract some energy from them. It is like a fuel for us. So when you eat carbohydrates, after complete combustion and oxidation of carbohydrates also, definitely there will be some byproducts or end products which are not useful for us. If you consume carbohydrates, you will be eliminating, giving out carbon dioxide and water molecules if it is complete combustion. If you eat lipids too, they also give you out carbon dioxide, water molecules. But in case if you digest, combust proteins and amino acids, guys, as we know that proteins and amino acids consist of carbon, hydrogen, oxygen, nitrogen and sulfur also. Along with carbon dioxide, water molecules, combustion of an amino acid will also give out nitrogenous wastes as well as sulfur wastes. Nitrogenous wastes may come out as ammonia, urea or uric acid, whereas sulfur waste may go out in the form of gas, or it may get mixed with your fecal matter too. So this chapter, this concept it was mainly dealing with elimination of combusted waste. So after combustion of food molecules, all those waste substances which are not required by your body anymore should be eliminated. Therefore, the process of elimination of nitrogenous wastes, what do you call this process? This is excretion, exit of unimportant, harmful, nitrogenous waste substances. Guys, from Porifera, first phylum of animal kingdom, to Chordata, most advanced of all phylums. Chordata, correct. In animal kingdom, Porifera to Chordata. Porifera were very simple living creatures without any tissues and organization, whereas in chordates we can see organ systems and many different types of sense organs, right? So as a move from Porifera to Chordata, definitely there was increase in complexity. Definitely there will be increase in complexity of every body system. Yes, there will be much better integumentary system in chordates, much better respiratory system, much better circulation and much better excretion also. So do not think that every animal will have same luxury of excreting same type of excreta. If animal is very simple, if it eats some proteins, it can just convert proteins into amino acids and amino acid into energy. But animal is so simple, it cannot make a harmless waste. If an animal is of very simple organization, the kind of excreta it will give out will be harmful, because it has no complicated organ system to convert harmful waste into harmless waste. So if you say like that, among animals, the most toxic excretory waste removed by animals from their body, and it requires highest amount of water also to dilute it. So what was that? Ammonia. So the most dangerous, most harmful excretory waste coming out of an animal body was ammonia. And animals which excrete ammonia were said to be ammonotellic. And this phenomenon of excretion of ammonia is ammonotellism. And this requires very, very, very high amount of water to be excreted, because it is very, very, very toxic. Okay, but in some animals like mammals and some sharks, marine fishes, and some land amphibians, we got good functioning of liver. Our liver will take up this ammonia, decrease toxic levels of ammonia. But how? Ammonia will be converted into urea. Urea is comparatively
less toxic. Therefore, there were animals which can convert ammonia to urea. They will excrete urea. So urea is mediocrely average, mediocre toxic waste, and animals which excrete urea were referred as ureotelic. And this phenomenon of excreting urea was referred as ureotelism. Done. And compared to ammonia, this may require just some amount of water to excrete, and it is not more toxic, average, mediocrely toxic. Toxicity. In case of arthropods, in case of reptiles, and in case of aves, these animals live in such conditions where water was very scarce for them. Water was not easily available. Therefore, their liver and kidneys have adapted a mechanism in which ammonia becomes urea, and urea becomes even more lesser toxic waste. What was that least toxic waste which animals excrete? That was uric acid. Yes. Now, animals which excrete uric acid were referred as uricotelic animals, and this phenomenon was referred as uricotelism. Uricotelism. And uric acid requires minimum amount of water to excrete, and it is least toxic among all these three. The more the toxicity, more the amount of water required to dilute it to excrete. Less the toxicity, less amount of water is required to excrete it. So, ammonia, urea, uric acid. Highly toxic, least toxic. More amount of water to excrete, less amount of water to excrete. Now, the question was, no animal will excrete their nitrogenous waste completely out of their bodies. Yes, they may get rid of 99.9% of nitrogenous waste, but a small quantity of it is necessary. They will retain in their body to maintain ionic balance, to maintain pH, acid-base balance, as well as to maintain molarity, osmolarity of body fluids also. So body is conscious enough, body was intelligent enough to know that how much must be excreted, how much must be retained. Even your body also consists of a pinch of urea. This urea in our body is very much important to maintain acid-base balance of your body pH, as well as this urea is also important for maintaining osmolarity. But guys, osmolarity means concentration. Hyper, hypo, isotonic. You might be aware of that, right? Hyper, more concentration. Hypo, less concentration. Iso, same concentration, isn't it? Some are required. Done? So among these, to which category do we belong to? Are we ammonotelic? Are we ureotelic? Are we uricotelic? Yes, tell me. Humans, mammals could be classified under which category of this three nitrogenous wastes? Are we ammonotelic, ureotelic, or uricotelic? Yes, we are ureotelic. Humans excrete urea. We excrete urea. We are ureotelic, ureotelic, okay, ureotelic. So do we have any organs for excretion of urea? Yes. Guys, the organ, tell me, there was a question for you. Tell me, name of organ which makes urea. Try this. Tell me, name of the organ which makes urea. Urea is made by which organ? Yes, very good, very good. Liver. Liver converts ammonia into urea. That's correct. Liver makes urea. And which organ expels it out of your body? Kidneys. Urea is manufactured by liver and urea will be excreted by kidneys. You should be very careful with this. Liver manufactures urea, kidney excretes urea. Both liver and kidneys will be communicating with each other. Liver will decide how much urea is made, kidney will decide how much urea must be excreted that day and how much urea should be retained in body. So if person has failed liver, you see, very often people who drink alcohol, they have got damaged kidney, damaged in liver, isn't it? People who drink more alcohol have got damaged livers. If a person was a very, very dangerous, deep chronic alcoholic, his liver will be damaged and he can not convert ammonia to urea, and such alcoholic people will have high amounts of ammonia still circulating in their blood, which will change their body pH. And if there is change in their body blood pH, enzymes and hormones and receptors does not work properly, isn't it? Guys, blood must be maintained slightly basic. Cytoplasm of animal cells was also slightly basic. And the foods we eat should be actually a basic or at least neutral. If you have this habit of eating acidic foods, body will be under constant stress to convert acidic food into basic and maintaining it. Okay, so try to eat basic alkali foods. Done. Now, so we have got liver and kidneys. Liver for conversion of ammonia into urea and kidneys for excretion of urea. So we are basically, fundamentally we were ureotelic. We do not require more amount of water, nor we depend on less amount. We require average, mediocre amount of water to excrete our urea, isn't it? Urea was acidic in nature. Your urine was acidic in nature. Only if you remove acidic urine from your body, body can maintain its alkaline nature. If you retain urea in body too much, body will become definitely acidic. Average. Okay, done? Now, so which organ for excretion of urea? Definitely kidneys. So we have got a system to collect urea, system to pass urine, organ to store urine, and organ to exit urine. Organs for filtration of blood, kidneys. Organs for passing of urine, ureters. Organ for storing urine, bladder. An organ for expelling this urine, urethra. A pair of kidneys, pair of ureters, one single bladder and one urethra. These four components comprise human excretory system. So guys, human excretory system consists of mainly of four physical components: a pair of kidneys, a pair of ureter, a single urinary bladder, and a single opening, that was urethra. A pair of kidneys, pair of ureters, single urinary bladder, elastic organ bladder, and urethra. These four components comprise human excretory system. TK or Kartikeya, sir, will you repeat all these things again in offline class? Yes. Yes, Kartikeya. See, online class, we are letting you know the fundamental aspects of a topic. It was not a complete information what you get here. We are letting you know what will you learn in classroom. It's like a trailer and main movie was waiting for you here, isn't it? Yes. Offline, if you come offline, everything in detail with pictures, with illustrations and animations. All of them are available because we are equipped with smart boards. Okay, I cannot show everything right now because it will kill some time for you. Okay. So, kidney. So, kidneys are bean shaped, reddish brown organs, isn't it? Each kidney kidneys are bean shaped, reddish brown organs. A kidney has both convex surface as well as concave surface, concave side. This was convex, this was concave. So, each kidney, it measures 10 to 12 centimeters, width 5 to 6 centimeters, and thickness of 2 to 3 centimeters. 10 to 12, 5 to 6, 2 to 3 centimeters on an average. And they may weigh 150, 160, 170 grams sometimes on a, because weight of kidney changes with an age and gender also, right? So average we have to consider always. And each has hilum. Okay. Now, so what was lagging? Video was lagging. Okay, that there was issue of a network. It happens. These network providers are not good enough. Same thing goes with Wi-Fi also. 4G, 3G, right? Not still 5G. That's okay, that's okay. It'll go slow, no issues. Okay. So this depression was referred as hilum. And this hilum opens into a funnel shaped structure known as pelvis. Done. Hilum opens into hilum opens into a funnel shaped structure referred as pelvis. Now, see this. Hilum into pelvis, right? Pelvis was again divided into many small parts So pelvis was again divided into many parts like this. Each part was called as a calyx. And as there were many calyx, so plural for calyx was calyces. Okay. So the exterior of a kidney was like a tough capsule. So exterior of kidney was like a tough capsule. And inside kidney, depending on color as well as depending on concentrations, molarities, it is very easy for us to split interior of a kidney into two parts. The outside, the surface of kidney like this. This looks light colored, hypotonic, not much concentrated, dilute. This part of a kidney, which is at the periphery, we can call this as kidney or renal cortex. Done? Guys, this was cortex. Done? Whereas the entire thing what you see in blackish color here, now red. This is interior part, we can refer that as renal medulla. And renal medulla was actually split into many pyramid-like structures. Those are triangular pyramid-like shaped structures referred as renal pyramids. Okay. These are renal pyramids. Okay. But you can see that medullary pyramids or renal pyramids. Guys, medulla is not continuous, right? There were some gaps between medullary pyramids. You can see clear, right? Isn't it? Medullary pyramids were not continuous things. Medullary renal pyramids. You can say medullary pyramid or you can say renal pyramid. They are confined to medulla. They were not continuous things. There was some space between pyramids. Therefore, even into this space, this renal cortex may dip in. So some part of cortex may also occupy some space between renal pyramids also like this. So cortex is also dipping into the medullary region like this. Like this. So this cortex, which is between pyramids, look like some columns. What do you call these columns of cortex which are adjacent to pyramids? We refer them as columns of Botany. Columns of Botany were extension of renal cortex into medulla. Columns of Botany were extensions of renal cortex into medulla. So that is. Now, this renal pelvis. See, guys, renal pelvis goes out as ureter. Isn't it? Now the thing was, at the junction of hilum, not only ureter, you guys can also witness entry of renal artery as well as exit of renal vein. Red was artery, blue was vein, because arteries mainly consist of oxy blood, therefore given reddish appearance, and veins usually consist of deoxy blood, therefore given blue images, blue color. Correct? Yes and yes, this is already taught, but all of you might not have attended, right? That's why we're repeating it. That's okay. Listen twice, that is much better for you. Done? So at the junction of hilum, you can see ureter coming out, entry of renal artery and exit of renal vein also. Both are possible. Both are there. Okay. So what was inside a kidney? Guys, kidneys have got this imminent capacity to filter your blood. Kidneys filter your blood. So per minute, at least 1100 to 1200 ml of blood is filtered in kidneys, which means actually your heart is pumping around 5000 ml of blood per minute. Of this 5000 ml, kidneys were actually filtering something around 1100 to 1200 ml of blood per minute. Isn't it? So 5000 ml blood was pumped by heart. It was referred as cardiac output. And of this 5 liters of blood, only 1000, 1100, 1200 ml is getting filtered, which means how much amount of blood is filtered by kidneys, which is sent by heart? 1/5 of heart pumped blood will be filtered every minute by kidneys. And by filtering this 1000 ml around, how much waste will be collected? If they filter 1100 to 1200 ml of blood per minute, approximately 125 ml of waste will be filtered. This filtrate was referred as glomerular filtration rate. The amount of filtrate made per each minute was referred as glomerular filtration rate. So if it is 125 ml per minute, how much per hour and how much in a day? Which will be approximately 180 liters per day. So 180 liters of filtrate was collected every day. 180 liters of filtrate, not blood, filtrate. But 99.9% of this filtrate will be reabsorbed because kidneys have done a mistake. Instead of filtering out useless substances, kidneys have also filtered out useful substances. Therefore, it is very important, it is as important to excrete urea and as important to retain useful substances too, isn't it? But the question was, who does this? who filtered blood inside kidney and who will get back, who will reabsorb this filtered back again. The special tissue or structures inside kidney, 1 million and more, which are mainly concerned with filtration of blood, collecting urine and reabsorbing useful substances and passing urine into ureters. What do you call the structures? Nephrons. Guys, we have nephrons for excretion. Every animal does not have same structure for excretion. Some animals like platyhelminthes, some cephalochordates possess flame cells, protonephridia. In case of askhelminthes, you can see rennet glands, solenocytes. In case of arthropods, you can come across Malpighian tubules, green glands. In case of Annelida, there were nephridia, isn't it? But in case of mammals, we got kidneys which are filled with nephrons. And nephron is collection of many epithelial connective tissue cells, and each nephron, it typically consists of a cup-like structure referred as Bowman's capsule, and followed by highly coiled tube-like structure and a U-shaped tube-like structure, and again highly coiled tube-like structure like this. This cup-like structure was referred as Bowman's capsule. Done. So this is Bowman's capsule. This is proximal convoluted tubule. This was distal convoluted tubule. This is collecting duct. All these things are more or less common in all nephrons, but less than 20% nephrons will also consist of a U-shaped structure referred as loop of Henle. Every nephron does not have loop of Henle. 15 to 20% nephrons will only have loop of Henle. We refer such nephrons as juxtamedullary nephrons. However, there are some nephrons which are like this. There is Bowman's capsule, PCT, loop of Henle, DCT, collectant, but they have minimal loop of Henle. Nephrons are also like, and for your surprise, more than 70% of our nephrons are like this, and they will not have loop of Henle too much, and these nephrons are referred as cortical nephrons. So we do possess two types of nephrons. Nephrons with loop of Henle's, juxtamedullary nephrons, nephrons without loop of Henle, that was cortical nephrons. Okay. See this. This is juxtamedullary nephron with loop of Henle. and this is cortical nephron without any loop of Henle's. Done. And blood vessel, artery, arteriole comes into Bowman's capsule, forms a network called as glomerulus, and exits out of Bowman's capsule, and again forms network around this, like this. Arteriole entering Bowman's capsule, afferent arteriole. Arteriole exiting Bowman's capsule, efferent arteriole. Efferent arteriole forms a network in PCT DCT loop of Henle, that was peritubular network, and one such network was found in loop of Henle. This was also referred as vasa recta. Okay, so Bowman's capsule along with glomerulus was referred as Malpighian body, or at least renal corpus. So blood gets filtered in Bowman's capsule, and that filtrate goes into PCT, loop of Henle, DCT, finally to collecting that. From collecting that, it goes into medullary pyramid, pelvis, ureters. So while filtrate is going from this point to this point, maximally body will try to reabsorb all useful things. Okay, that was referred as reabsorption. So today we learned again basic things about kidneys and nephron. How does that look? And once you come here, we will also discuss about actual mechanism behind nephron filtration, reabsorption, and also neural control of urination, which is called as micturition. Done. And how our urination could be controlled hormonally and neurally, both are there. So once you come here, we will discuss them. Okay, shall we sign off? We'll meet tomorrow. Thank you.